Hello everyone. Thank you for joining this new session after the summer break of the BioClock Academy lecture series. We are back with a fresh start and with new and exciting speakers also outside of our bubble, outside of the BioClock consortium. Happy to see many familiar but also new faces, or I would say names here. My name is Juana Georgiana Rus Oswald. I am a postdoctoral researcher in the Dutch BioClock Consortium, and together with Hannah Brooks, who is a PhD student in the same consortium, we will host the session today. For the ones who are very new, just a brief introduction of who we are and where are you located. We are within the BioClock Consortium. This is a Dutch research organization which was set up to study different aspects of the biological clock in different pillars, so in humans, in health, and in society. And the BioClock Academy is a lecture series organized within this consortium, and it is addressed to anyone who is generally interested in the field of chronobiology, but especially addressed to early career researchers. And its aim is to introduce you to the basic concepts and equip you with the proper theoretical knowledge by experts in the field. This lecture series takes part every third Wednesday of the month, so maybe put it as a fixed thing in your calendar. It's actually always exciting. Or just register for the talks, then you have it for sure there. And the time frame for today will be the following. We will have a 40 minutes talk, followed by 15 minutes of discussion, so roughly 1 hour, so that we can close then at 5 p.m., so our time. Our speaker today took the early birds benefits into account, and she sacrifices her early morning to enlighten our afternoon. During the talk, you can also write your questions in the chat so that we can discuss them later. And now I will give the floor to Hannah, who will introduce you to our guest speaker today.
Yes, so also a warm welcome from me. Today I have the pleasure to introduce you to Dr. Emily Manoogian, who is a postdoctoral researcher at the Salk Institute in San Diego. Here she works in Dr. Panda's lab, where she actually also serves as head of the human studies. So the focus of her research is mainly on the timing of different behaviors, such as eating, sleeping, light exposure, activity, and to look into the health impacts of those timings. So she's also a true advocate for the field of chronobiology, so it's a real pleasure to have her here speak today. And the subject of today is time-restricted feeding. Like Georgiana said, it's very early morning still, so I hope we didn't mess up all your timings. So I don't want to let you wait any longer. Please, Emily, the floor is yours.
Thank you so much for having me, and thank you everyone for coming to this. I realize it's not quite as early for you guys. It's middle afternoon. It's just 7 a.m. here. But I think this will be quite a fun talk. So thank you for having me. It's a really cool seminar series that you have. I was looking at the lineup, and it's quite an impressive bunch, so I'm happy to be a part of it. All right. So health effects of time-restricted eating. We'll get to a lot of different aspects of this, and there's so many components of what's come out in this field in the past, 5-10 years. This field has really kind of exploded. So we'll touch on as much as we can, but I am really excited about, a longer discussion. So if I don't touch on what you're interested in today, please, let's talk about it. So just to give some context, and I probably don't need to say it to this group, but I like to just kind of give us all the same context that we're working with. So we all know our body is filled with endogenous rhythms created by molecular clocks and internal networks that work together to combine and output this, rhythmic output in pretty much anything that you can measure, from behavior, anything from when you're awake to when you're asleep to what your mood is and your cognitive abilities, your physiology. I like to say anything you've ever had measured at the doctor's office probably has a rhythm, and any even individual cell function. And we don't live in a cave, although it would still work if we did. We have all these natural 24-hour cues, 3 of the best known being light, dietary intake, and activity. And it does not just work in a linear fashion. This all kind of feeds back on itself, right? So when we're awake dictates when we get light and when we eat and when we're active. And how we're feeling probably also dictates when we're active and when we eat and when we get light. And all of that's just from the behavioral point, but this physiology all feeds into that. So it's an interesting feedback loop where it really combines these endogenous rhythms with these external outputs. And we misalign any of those in external outputs with what our body is trying to do, we can have disruption in these rhythms. And it's always nice to keep in mind why we have these. It's an anticipatory system. It's really preparing your body for what it needs to do and also separating incompatible events. And I think that's a big thing, actually. When we talk about time-restricted eating, I think that's a big part of it. And we can get into that more a little later. And really, if you kind of oversimplify it, it's really keeping everything in the right place at the right time. And if we don't do that, this was a figure that we put together a few years ago for a review, really looking at all of the diseases, chronic diseases that are really associated with circadian disruption. And kind of you see different prevalences at different ages, and there are different severities. So anything from a very mild, you're irritable because you're jet lagged, or you pulled an all-nighter, or you're just really kind of out of sync, to everything up to heart disease and Alzheimer's and Huntington's. And we've seen actually in a lot of rodent models that even some of these diseases can be directly improved, symptoms can be improved just by a few circadian interventions, some of which were time-restricted feeding. And so today we're going to be talking about this one part, the dietary intake part. And obviously light and activity are also important, but time-restricted eating is really just trying to get at this one core component about how we affect our clocks from our external cues. And I'm going to be talking about time-restricted eating today specifically, but I think it's important to note that when we talk about temporal patterns of eating, or temporal eating patterns, or circadian optimization through dietary intake, there's a few ways that you can do it. And time-restricted eating takes multiple of these into account, and it can take all of them into account, and they can be combined. But there's no really one like fully circadian optimized eating pattern. And really, because there's a lot of different components to it. And based on where you are, currently, there might be one or two or three different sections that might be more important to you. And there's probably some you're already doing. So very briefly, duration, the timing for each day that you're eating from your first to last calorie is important. And you see that being addressed not just in time-restricted eating, but other forms of intermittent fasting. The phase, so when in the day are you eating? Is it something really in the early morning, right when you wake up? Are you waiting half of the day until you have your first meal? Are you eating just before you go to bed? The timing we know is very important, and that's usually what gets lost in other forms of intermittent fasting or general fasting interventions. Variability, which I think is highly underrated, both for when you eat as well as when you sleep, which we can talk about more later as well. But that day-to-day change from when you're taking in your calories, especially first and last calories. And then frequency, being how many times are you eating? And there are some interventions where they put you on just 3 meals or 6 meals or more. And the research on frequency is a little bit more mixed and a little bit more nuanced. But today we're just going to talk about TRE, but I think it's important to keep those things in mind when you're reading about this type of literature and when you're conceptualizing, like, how could we make changes or what's really happening? There's really a lot of components to it. So just to set a definition, time-restricted eating is really a dietary regimen in which all caloric intake is restricted to a consistent, and that's a key part of TRE, a consistent period of the 24-hour day, resulting in anywhere between a 12 to 18-hour fast. So that means typically you're eating somewhere between 8 to 10 hours in a day. So for instance, like 10 to 6 or 9 to 7, something like that. And there's no explicit limit on energy intake. So there's no required caloric restriction, although in human studies, we do find that typically there is some indirect caloric restriction because people tend to cut out late night snacks. And the difference between TRE and TRF, TRE is time-restricted eating. TRF is time-restricted feeding. It all started with feeding, and then we started telling humans that they are feeding and they didn't like it, so we switched it to eating. So typically now if you see TRE, it's humans, and TRF is animal studies. All right, so what are the origins of time-restricted eating? So it all started with mice, of course, and this links back quite a while now. It originally started from data from Uli Schibler's lab that showed that when you restrict feeding, you can actually uncouple these circadian oscillators in both the central and peripheral pacemakers from the SCN. And that was really exciting to see that you were able to kind of make this, pull these clocks apart by feeding. And that's partially because food does not directly affect the clocks within the SCN. Things like light are going to have a much bigger effect on that. But we know that dietary intake does directly influence the clocks in the periphery. And this was really the first paper to show that. Not too long after, Michael Miniker's group showed that you could entrain circadian clocks in the liver by feeding. So not just daytime feeding or nighttime feeding, but he could actually show how they would phase shift over time. And different organs shifted at different paces. So this was also really interesting. Next was this kind of, I think, really a key experiment from Joe Bass's group that showed that when you put animals on a high-fat diet, of course, we know that this is how you create diet-induced obesity in a mouse model, and this is how they gain weight. But they also found that they're not just gaining weight, they're also changing the pattern of when they eat. And so in these mice, mice usually eat at night when they're awake. They will sometimes have little snacks. And you can see these on the left here. You can see that in the feeding behavior, if you look at on the left, you can see there's little snacks going on during the daytime, but it's quite limited. Whereas when you look at the high fat content food, you can see that they actually lose a lot of that rhythm and they start eating a lot more during their day. It seems like they're just eating kind of around the clock. And then from Sachin's lab, he also found, as before I joined the lab, but he was also able to show that there were hepatic rhythms and gene expression that were also driven by food, which is right in line with that, and seeing this is happening at a gene transcription level. And really, whatever you go back to, whatever system you look at, you can see that they're somehow affected by timing of eating, whether it's modulation of protein synthesis or cholesterol metabolism. Almost every aspect of metabolism is affected by when you eat, which is really, really interesting. And this led to the very simple experiment that Sachin's team did, wow, quite a while ago now, almost 20 years ago now, where you took this very simple question. We know that when you put animals on a high-fat diet, they gain weight. We also know that they start eating at all times of day. And so the question is, what if you only let them eat during the night? Would they still gain that same amount of weight? So there was an ad lib group, and there was a group that could only eat anywhere from 8 to 12 hours was tested. And after 26 weeks, they found that the mice that were on ad lib gained in weight and became obese as they would have expected. But the mice that ate the same amount of high-fat food, had the same exact dietary intake, just at a different time of day, did not gain weight. And they gained, I mean, some weight, but they looked like a normal mouse, like they had been on normal chow. And what was also really exciting was the mice that were ad lib ended up developing fatty liver disease, whereas the mice that were on the time-restricted feeding schedule had a healthy liver, even on that same high-fat diet. And that was really striking data, which had led to many, 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 many studies happening since then. Oops, sorry. And I don't have time to go through all of those today, but many labs around the world have done research on time-restricted feeding now, looking at many different outcomes, looking at many different genetic models of mice. Some that are a lot started in the metabolic realm, but now there's a lot in cancers, in cognitive decline or neurological diseases like Alzheimer's and Huntington's, seeing these really widespread improvements. And so many different things have now been assessed to see that time-restricted feeding has really systemic benefits on an animal. But then we get to humans. So what about time-restricted eating in humans? So to kind of say, what does this look like? So a few common time-restricted eating windows you've probably heard of. Sometimes there's like a 6 a.m. to a 2 p.m. or a 12 p.m. to an 8 p.m. These have kind of commonly been termed as early time-restricted eating or late time-restricted eating, again, trying to get at that component of phase. Unfortunately, for a lot of people, this is either too early or too late. If you're sleeping from 10:30 to 6:30 in the morning, having your eating window start at 6 a.m. doesn't make any sense. You would actually miss part of it. And also, we think it's important to not eat for a little bit after you wake up to let melatonin decrease, because melatonin directly suppresses insulin secretion. And so you're not able to actually process food and take glucose out of your blood properly until melatonin has gone down. So we think early time restricted eating, if you just set a clock on the wall, probably doesn't make sense. 6 a.m. to 2 p.m. may be great for some schedules, but for what we usually think of as a typical sleeping pattern, it's probably too early. And then some are too late. 12 to 8 became very popular, kind of through the media, I think. And again, I think this probably leads to eating too close to bedtime and probably having an unfeasible fast in the morning. But again, depending on your schedule, that could be the right time for you. So the way that we do it within our team is we do a personalized time restricted eating. So it's really 4 steps to figure out what your time restricted eating window should be, and anyone could do this. So the first thing you do is you set when you sleep. So we'll take an example of 10:30 to 6:30. Then you'd say wait at least 1 hour after you wake up and wait at least 3 hours before you go to bed. This will allow melatonin to come down. And then also before you go to bed, we know it's important to get those glucose levels down before you go to sleep. And there's some really beautiful work done by Frank Shear's group that's shown that when you have a high glucose load, not too long before bed, and the glucose is still high when you go to sleep, that it stays high. And again, this is where we get back to that separating incompatible events, where you need to make sure that your insulin is able to be high and process glucose, and that you're not trying to, your body's not trying to maintain glucose in the blood when it's still high and hasn't had a chance to go down yet. Next, you want to identify any important meals. So say you have a family dinner at 6 p.m. that you need to get to, and you're like, this is important. You want to make sure to include that. And then you really kind of count backwards. So you could say 6:30 is the earliest I could stop. So I'll go 10:30 to 6:30, and that would be an example for an 8-hour, and that would provide 4 hours after you wake up, 4 hours before you go to bed. Or you could do a 10-hour, which I think is plenty long. Which still gives you 2 hours after you wake up and 4 hours before you go to bed. So for someone who's sleeping from 10:30 to 6:30, this is likely one of the ideal patterns, but anywhere within that dotted line, you could move that eating window, and biologically, you should still be good, at least based on what we know so far. So how do we actually do this in humans? So it's a little bit tricky because traditional methods for assessing dietary intake really don't capture many aspects of temporal eating patterns. So before this whole field kind of started, common tools were like food frequency questionnaires, which do not ask about timing, 24-hour dietary recalls, which are very informative, but they're only off of one day. And from our work, we know that there's huge amounts of variability in both the timing and quality and everything really that has to do with food. There's common or weekly food journals. Unfortunately, there's a lot of people don't always do them in time. And so they'll go back and log after the fact. We know we have a very poor memory on when exactly we ate. And this leads to having really either very short periods of logging that are frequently filled out retroactively. So there's issues with that. And they really don't capture the variation in first and last calorie or eating duration or the timing of eating. They're also not incorporating sleep. So you really don't know what the phase is. And so our lab has created a app called My Circadian Clock that tries to get at a lot of these issues. So it tries to decrease logging burden because you can take a quick picture. It saves things you've named before so you can easily click on it. It's convenient because you've always got your phone on you, and that's true for most people now. It also allows for real-time monitoring of participants. So we're able to see everything that's logged in real time on a coordinator portal on our end. So if we're monitoring participants, we can see if they're logging, we can see if they're doing things retroactively or not, and we can give them little pushes or little reminders to be able to, kind of help them stay on track. We can capture multiple types of data at once with this. So anything from food, sleep, exercise, health factors, we can send out questionnaires. So it's very powerful, kind of a one-stop shop for that. And it's really a good way not just to monitor interventions, but to also implement interventions. So as I said, you can send reminders, but you can also send scheduled reminders like, hey, you're on this week of the study, keep it up, or you have a visit coming up. And we can also send educational information, which is really powerful. And it's nice, this is scalable, and we can do this remotely. And we've collaborated internationally doing this. I see some people that we've worked with previously. on the call now, which is just fun. But it's really been a powerful tool in our research. So luckily, if you ask people to take pictures of their food, they do. These are a few examples of some of the food items that participants have sent in over the years. And what we do is we take those items and we create what we call a feedogram. It's really a raster plot of all these eating events where you just line them up over a 24-hour day. So each line that you're going to see is one day with each eating event noted as a little tick mark. But we don't just do it for one day. Typically, the shortest duration that we do any type of assessment is about 14 days. And that allows you to see patterns in eating. You can see in this individual, you can easily see on weekends, they're eating at different times. You might want to break down what's happening between 6 p.m. and 6 a.m. And you can collapse it all into one day. So you could say in these 2 weeks, this is when your body kind of saw food. You can turn it into a circle. So this is a 24-hour plot. So you can say on one 24-hour day, these are all the times that someone ate. We don't just do it for one person. This was done in 156 people in San Diego. And you can kind of see people are eating all around the clock, anywhere from really 5 a.m. until midnight. It's pretty strong but there's still eating events throughout, with 3 a.m. being kind of the low point in our area. And as we talk to nutritionists, their saying was, if your eyes are open, your mouth is open, which we found to kind of be true. People just really like eating, and they do it for many different reasons, and hunger is probably not the main one. And in that study, they found that over 50% of adults in our area eat for over 15 hours a day, which is a huge amount of time. And it really kind of gave this first glimpse into knowing that this was going to be an important field and that there was really a lot of room for improvement among humans in the modern day. A couple things I want to really point out about important considerations to take when you're looking at time-restricted eating studies, and really thinking about how to measure this data. So as I said, 2 weeks is probably an ideal amount of time, and you could definitely do longer, get more precise, but 2 weeks seems to be a good amount of time to understand when people are eating. This allows you to capture weekends and weekdays, which can be very different in individuals. Really, it's off days and work days, but for most people that works to weekday. So a minimum of 1 week, I'd say, is necessary to really understand those eating patterns, but 2 is really better because you see a lot more variation. And if someone doesn't log, you have another chance to capture it. Ideally, you're combining food and sleep information together. And additional measurements can be really important depending on your question. So for instance, if you're really interested in sleep, obviously tracking sleep timing is important, but you might want more specific measurements. If you're really interested in tracking glucose regulation, then a continuous glucose monitor is also probably really helpful for you. But it depends on the question you're asking. But I think a minimum is really capturing the food and sleep. And there's many different tools to do this. Many of you are probably familiar with these ActiWatches. There's just a few examples. The Philips ActiWatch was really commonly used for a long time, but has now gone out of, they've kind of discontinued it. So now people are moving more to Condors or Garmin's or Fitbits, and there's many other watches that can be used. Oura rings are also very popular now, and especially in just kind of consumer products. But these can capture many different things, more than just sleep estimations based off activity. They can also capture light, which is really important, which is something that most of the commercial products are lacking. But they can capture heart rate and body temperature, and they can be really powerful tools. So to give you a few examples of what we're looking at here. So here's an example of 2 weeks of food logging. Each little orange dot here is a caloric event that was logged. And the kind of bars that go around it are just kind of summing up from first to last for that individual day. And we do not just look at when people, like the duration for any given day. So one of the things that our lab does is, rather than just looking at your daily eating duration, which you can see on the right side here, varies very greatly from day to day for this individual. The average is about 13 hours, but it goes anywhere from 8.5 hours to almost like 14.5 hours. Or actually, there's a 17-hour day there. We don't just do that. We actually look at a 95% interval. So what we do is, when you look at the 95% interval, is you take away the earliest 2.5% of events and the latest 2.5% of events. You can call those outliers, and you can see those being avoided here. but you can say this is kind of the window that someone eats. And we think that's really important because, as noted on the top here, there's a lot of variability in both first and last calorie. And this participant alone, we saw they had their first calorie anywhere from just before 6 a.m. all the way to 11 a.m. and their last calorie, again, just before 6 p.m. and leading almost up to 11 p.m. And when you look at this interval, we could say 95% of their caloric intake happened between 6:30 and 8:47, which is just over 14 hours. And again, this is really important because you can see all these dramatic differences. If you just look at 3 individual days, you can see they range from 8.5 hours to 11 to 13.5. And when you just take a glance, they don't look all that crazy different. But when you're really looking at how the body is seeing food and the cues that you're giving it, they're quite dramatically different. Then, as I said, it's important to layer on sleep. So these are a few examples of the wake and sleep onset for that individual. And you can also see that there's really large gaps here between when people are finishing their food and when they're going to sleep. So in those same 3 days, you can see the time from wake to first calorie on the first day was over 5 hours, whereas on subsequent days was either 2 hours or 26 minutes. And then the same for when they ate their last calorie to when they went to bed, ranged from anywhere from 45-minute interval to over 5 hours. So there's this huge variability, not just in the timing of eating, not just in the timing of sleep, but this combination of when you're eating relative to your sleep. And so again, I think it is important to look at these kind of 95% intervals. I think it's really important to also look at the average eating window to see kind of what you're working with. You know, are they consistent on their average? Are they moving around? But variability is really a key component here that I think is often still ignored. And I think we'll see a lot from this moving forward in this field. One really exciting thing that we saw in one of our pilot studies that we started with a few years ago now was that time-restricted eating also decreased the variation in timing in the first and last calorie and wake. So it's not surprising that you decrease the timing, the variation in when you eat, right? If you set an 8 or 10-hour window, you're probably going to be eating on the borders of those. But what was exciting was we also saw a significant decrease in the time, in the variability of when people woke up. And so people were becoming more regular sleepers as well. And we saw a trend in the onset of sleep, but it was actually statistically significant for wake, which was really kind of interesting. And I think, again, will require further follow-up. So when moving into clinical trials, we see that there's a need. We have these tools for it. We know the things to consider. But what really are we asking here? So the first questions were, is time-restricted eating feasible for humans? Is it safe? What time should you do it? What's the optimal duration? You know, who is this going to be beneficial for? What are the benefits? And what are physiological mechanisms? And as I mentioned earlier, in the past 5 years, this field has really exploded. It went from, 5 to 10 studies to now there's over 100 studies on this. And we're not going to cover all of that today by any means. So I recognize that I'm ignoring a huge amount of the field. But time-restricted eating really impacts what we've seen so far. Most studies have focused on individuals with cardiometabolic disorders. Many of them have studied on individuals that have elevated BMI or obesity and looking at it as a weight loss mechanism. We do see that typically there's about a 3 to 5% weight loss that cannot all be explained by changes in calorie intake. But more importantly, I think it's actually affecting other parts of metabolism and cardiometabolic health. The results that you'll see from any one study really depend on what their levels were at baseline, and what the intervention really was. So in many studies that gave null results on changes in A1C, which is like kind of this gold standard measurement for understanding average glucose over approximately the past 3 months, those people had normal levels to begin with. Even in our own research in shift workers, if they were healthy to begin with, they stayed healthy. You don't see a significant shift in that, and I think that's to be expected. And so it's important to consider who your population is going into it. But across many different studies, there's been a few trends in aspects of, what you can call metabolic syndrome, which is really having any 3 of these 5 factors listed here. And I think it's been pretty consistent across the board that many groups have either seen null results or improvements in weight, glucose regulation, blood pressure, cholesterol, and triglycerides really has been a mixed bag, which it gets a little bit more complicated and probably relies a little bit more on the individual's diet than just the timing. And again, the factors I think you need to consider whenever you're reading a TRE study is the population, sex, age, health status, their chronotype even, study designs, what their eating windows were at baseline. Unfortunately, especially in the beginning, but still now, eating window at baseline is not always assessed. And people get put on an intervention, but you don't know where they started to begin with. And that also, it's kind of like saying, we're going to do caloric restriction, but we don't know if you're already doing caloric restriction, which might be an issue. So I think assessing that at baseline is, really important. The phase of the eating window that's being used, the study duration, and also adherence is really important. Not all studies are created equal, and some of them are not having the same kind of monitoring or logging or tracking and aren't really able to get a good gauge on adherence. Okay, so with all of that in mind, I want to tell you a little bit about one of our studies that we did in shift workers, and really why this is so important. So shift workers, we know, have an increased risk of chronic disease. And over 25% of the workforce does shift work. It's associated with many chronic diseases, including cancer, diabetes, cardiovascular disease, affective disorders, things like depression, anxiety. They're all associated with shift work and circadian disruption. We know that shift work is considered a carcinogen by the World Health Organization. And unfortunately, shift workers are typically excluded from clinical trials. I'm guilty myself. Shift work is a huge confound. So when you're trying to assess something in humans, you don't want to put them, have this huge differentiating factor, which is why we really have to have specific studies to be able to study these shift workers, because they really probably need the most circadian help of all of us. Just some data taken from the MCC app, which has been not published yet, but we found that, late night and erratic eating patterns are really common among night shift workers, which is not surprising. They're eating while they're working those night shifts. So these are those same kind of plots I showed you before. And you can see on the right are the regular workers that had reported on the MCC app. And you can see they show that same pattern I showed you from before. But among shift workers, you see this kind of really, there's no, maybe between midnight and 6, it's a little lighter, but you're really seeing eating all around the clock. It's also been shown that shift work increases rates of obesity and compromises glucose regulation. This is also really interesting because this kind of goes against the calorie in, calorie out model a little bit, in that a lot of these shift workers actually have much higher rates of physical activity and lower sedentary behavior than most others. You could think about first responders, or, like a nurse working through the night is a very active person. They are not just sitting there in an office like we are during the day a lot of the time. They are moving. And despite being more active, they're still gaining higher rates. And there's been an association between the work night total food intake being associated with cardiometabolic syndrome. So not just how much you eat, but when you're eating it has been shown to be associated with cardiometabolic disorders. And we really think this misaligned eating duration really is probably what's leading to this compromised glucose. And again, there's been really nice work in shift work simulated in labs to show that eating during the night leads to misalignments of glucose and insulin, which can lead to really maintained high levels of glucose through the evening. So our first research study into this foray was called the Healthy Heroes Study. This is a collaboration between UC San Diego, the Salk Institute, where I'm based, and the San Diego Fire and Rescue Department. And it was a 3-month randomized control trial with follow-ups every 3 months through 1 year. And it was 2 groups. Participants either randomized to what we consider a standard of care, which was really a Mediterranean diet, or a standard of care plus a 10-hour time restricted eating. And we did this in 150 participants, both male and female, in San Diego. So why firefighters? A majority of firefighters work 24-hour shift increments, but they can do this up to 4 days in a row. Now, they are allowed to sleep at night, but they get called multiple times throughout the night and have to be able to jump up very quickly, get to the truck, and be on call. And so the quality of sleep that they get on those nights is also very poor, and some people do snack in between those calls. It's the most common cause of death in firefighters actually is not cancer, it's heart attacks or heart events, so strokes, heart attacks later on. And like I said, only about 0.01% of clinical trials are actually studying shift workers. So we knew this was something that needed to be addressed. And this was really our first look at TRE in shift workers in the real world. And so our first questions were, is it feasible? And how could we impact their cardiometabolic health? We actually did 24-hour ride-alongs to test this out with them, which was exhausting, but very enlightening. And it did help us shape our study design before we finalized it. So just quick overview, this participants start, we were able to recruit them. They come in for their first visit. They get trained on how to use the MCC app. We take their weight, vitals. We put on a continuous glucose monitor, which assesses their glucose every 15 minutes for 14 days. That they're just able to wear passively. They also put on an ActiWatch. And for 2 weeks, we tell them to go live their life. This is how we collect our baseline measurements and also screen out for any exclusion criteria. At the second visit, we do a 24-hour recall with the dietitian and give them their randomization group. And then we track them for the next few weeks. They get a call in the middle of that intervention. And then at the last 2 weeks, they come back in to put on, we actually went to the fire stations to give them their CGM and watch, wear that for 2 more weeks. And then at the very end, we'd repeat those measurements, and we repeated those every 3 months. So a quick look at what these look like. The MCC app, which I already showed you, a Philips ActiWatch, and then this, we use the Abbott Freestyle Libre Pro for this study, which is a small continuous glucose monitor that you just wear on the back of your arm. And I'm going to skip through this a little bit because I'm taking a long time, but this led to huge amounts of data production on many different things, which allows us to create really cool figures like this, where we're able to combine the top, you can see all the food entries from the app. In the middle, you can see the glucose on a given day. This is all from one individual day from one participant. You can see how it's changing across the day. And then you can see the food items above that are tracked to where it is on when it was consumed on the glucose monitor. And then the bottom, you can see when that person was active, when they were receiving light. And you can kind of combine this to create these very personalized windows, which is also a very powerful tool for educational tools for participants. We found that TRE was feasible. So just to orient you, these are the same kind of graphs, but they're inlaid. So the inside circle is baseline. You could see participants in both groups ate around the same time at baseline, but then the outside is the end of the intervention, that 3 months, you can see the participants in TRE were able to delay their food, their first food, and advance their last food. So we're not meal skipping here. We are trying to just kind of push the food in a little bit on each side. And we saw that they were able to decrease their eating window by about 3 hours that way on average. In this population, we did have most vitals and health assessments were normal when you look at the full group, but we did see that cardiometabolic risk factors did exist in different amounts. So the largest thing that we saw was LDL cholesterol was elevated. BMI was also high, which can be inaccurate in such a active population. C-reactive protein, which is a marker of inflammation, and then blood pressure, glucose, triglycerides. Many of these metabolic factors had at least a few people that had these issues. And when we looked at that group that specifically had those health deficits at baseline, then we saw significant benefits in VLDL particle size, which is very low density lipoprotein. So these are, kind of think of it as the worst, it's what LDL progresses to, which can lead to atherosclerotic plaque. So it was exciting to see that decrease. We also saw benefits in quality of life assessments for emotional well-being. HbA1c, which again is kind of the gold standard for glucose, improved, and diastolic blood pressure also improved. And just looking at A1c, just to kind of highlight this importance of looking at the proper group, in the left, you can see that was the change for everyone, which you could say is almost nothing. Whereas when you looked at the individuals who had elevated levels at baseline, with the standard of care on the left and the time-restricted eating on the right, you can see that the standard of care got a little bit worse or very mildly better. But in the TRE group, we saw benefits at every 3 months throughout the study. And one more exciting thing on that was we also saw improvements in LPIR, which is lipoprotein insulin resistance. And this was in the full group of participants. And so this may indicate that there could be preventative measures moving forward, and this requires further follow-up. And just to give kind of a little brief assessment of what the continuous glucose monitors look like, this is 2 examples from time-restricted eating participants. On the top, you can see their average over those 14 days, with the darkest line being the median. You're looking at the confidence interval around it. Baseline is in yellow, so you can see these are really high levels, whereas the blue is just at that 3-month time point. You could see on the left, it just completely kind of comes down. So the levels are going back down to normal levels where they were elevated before. And on the right, where this person had much more variability, you really see that they start having lower levels at night. You can see this first, I don't know if you can see my pointer, the first half of the day over here is really decreased there. And you see that they're having a more normal peak during the middle of their day, and they're able to come down at night. So in summary, long duration eating is common in modern society. Over 50% of adults do it. We know that it's common in shift workers. We know that it leads to a lot of different issues, and time-restricted eating may be very helpful in this realm, but really larger multi-site clinical trials are needed. And the one last thing I will leave you with is shift worker schedules are not limited to shift workers. So many adults that do not call themselves shift workers have a job that requires them to be on call 24/7. When called, they might need to be active for 30 minutes to 4 hours. They might only get to sleep for a few hours at a time. And it's always going to be on for at least 4 hours between 10 p.m. and 6 a.m. So this is actually data from myself. When I had this happen to me, this was my normal work schedule. You can see sleep is highlighted in blue here. The yellow is just light exposure. And then this was my shift schedule. And you can see that my sleep is completely desynchronized. It's all over the place. It's very random. And you would say, what were you doing to have such a horrible rhythm? And I'm guessing some of you can guess this, but it happened on that day, and I had a baby. And many people go through this type of short-term, but short-term being months of this almost no rhythms, especially if you're the mother who's breastfeeding every couple of hours. This is extremely challenging on your body and your mental health, and it is something that we're all facing, and there's really not much to say to help people right now. And this is another group that I think really could use any circadian help they could get. And yeah, one other time that we all circadian disrupt ourselves is when we travel to conferences. So many of you might be familiar with SRBR. I was very lucky to run this with a good friend of mine, Anlar Huber, this year. At this SRBR, we ran this study called Disrupt. We're starting to assess when people are changing their eating patterns when they're sleeping, and how both the travel to the conference and the conference disruption itself is probably causing a lot of different issues. We're going through the data now, but I think it's something to keep in mind that even if you don't have a baby and you don't do shift work, I bet you've traveled to a conference and you've probably had at least short-term circadian disruption as well. So with that, I want to give some acknowledgements. We could not have done this firefighter study without the help of San Diego Fire and Rescue. Specifically, Dave Piccone and Kyle O'Neill were really amazing champions, and our long-term collaborator, Pam Taub at UCSD, really crucial in all of this. And with that, I will acknowledge the rest of the lab. The people in bolded are ones that actually worked on the firefighter project. And again, they were all amazing and crucial in all of this. So thanks to them. And with that, I will open it up for any questions.
Well, thank you very much, Emily. Very interesting. A lot of new information for me, actually.